Everyone, we're from Group F. Our topic is genetically modified organisms, so-called GMO. I'm Kevin Tan and I'm going to define the definitions of GMO. Define the definitions of GMO. Genes from a genome of one species can be extracted and artificially forced into genome of an unrelated plant or animals. These genes can come from bacteria, viruses, insects, animal, or even human. The plant or animal that receives these genes is called genetically modified organisms. They are also called transgenic organisms because of the transfer of the gene. Now I'm going to talk about some history of the GMO. In 1973, the recombinant DNA created. The idea of man-made DNA or rDNA came from a graduate student at Stanford University Medical School. Further, in 1982, the FDA approved first GMO. Human insulin produced by genetically engineered E. coli bacteria appears on the market. Further, in 1994, GMO hits the grocery store. The US Food and Drug Administration approved the flavorless Savour tomato for sales on grocery store shelf. The delayed ripening tomato has a longer shelf life than conventional tomatoes.
Hello, I'm Li Yun. I will continue about the parts of process of creating GMO. GMO are created using biotechnology through genetic engineering, which is also known as genetic modification. This technique is completely different from traditional breeding as it involves transfer of genes from different species. This technique cannot occur in nature. DNA has to be isolated and inserted into the vector and transformed into the host organism. Living organisms have natural barriers to protect themselves against DNA of other species. Therefore, this technique is quite new and quite complicated. Scientists have been working on some interesting combinations through this technique. There are 4 steps in genetic engineering. First is to identify a trait of interest. Number 2, isolate the gene for the genetic trait of interest. Comparative analysis of genome is undergone to identify the gene for the trait. Step 3. Gene is cut and pasted into the plasmid using an enzyme, and the plasmid is inserted into the bacteria that will infect the target cell. And lastly, after the gene is successfully inserted, the GMO is carefully controlled to grow in growth chamber, and the growth are monitored to ensure that the new GMO grow and replicate carefully. After the GMO successfully replicate and a new GMO is created.
My name is Li Keilan. So now I'm going to talk about the pros of GMOs. So in agriculture, GMOs increase the yield and the quality of the crops, reduce the food waste, and also increase the pest and herbicide resistance in the plants. Besides, it also helps to strengthen the trunk. So in medicine industry, GMO helps to produce edible vaccine and antibiotics such as insulin. And also, it helps to produce organs for transplantation and also reduce transmission of disease. So next, for the environment, GMO help to remove the heavy metal such as copper and also act as a biosensors. Besides, it also takes an important role in population control and increase the survival rate in harsh conditions. So next is for the cons of GMO. It includes gene escape, allergy risks, increase of use of pesticides, antibiotic-resistant bacteria, and also this can lead to pests can adapt to the pesticide and herbicide. So next, it also can lead to cancers, infertilities, gluten disorder, pregnancy, and toxicity of insects, and this can cause pollination problems.
Hello, my name is Kevin Kuwumping. So when we talk about GMO, we talk about the successments of GMO. So what have GMO achieved in our life and what is the benefits it brings to us? First of all, when we talk about plants, golden rice is a successful example of GMO. It is first announced in year 2000. The golden rice is a rice that is rich in beta-carotenes, unlike usual rights, and it helps to improve human health. It plays a very important role in the ongoing global efforts to fight against the vitamin A deficiency in poor countries. So when we talk about achievements in GMO, we must talk about the achievement in animals that is modified. And here we have two examples. One is eel mouse and fast-growing salmons. Eel mouse is one of the successful projects in medicine. Through genetic engineering, a human ears could be grown on the epidermis of mouse. This discovery may lead to the growing of human organs on the other animals for transplantations in humans. Besides, for fast-growing salmons, genetic modified salmons take shorter time to reach maturities and be ready to consume than natural salmon. Besides, when we talk about microorganisms, this is very familiar example by which insulin is made from bacteria. A genetic modified E. coli bacteria able to produce human insulin in vast quantity at a higher production rates did result in lower insulin price, and which is a greater benefits for patients.
Hi, I'm Kevin, and I'm going to make the conclusions of our team about GMOs. Our team agree on the benefits of GMO for humans, but there are some restricted boundaries to reduce its risks. For example, in New Zealand, the importations, development, testing, or release of GMO are strictly regulated. Such activity must be approved by Environmental Protection Authority. GM techniques have been approved for use in research involving both plants and animals, objects to various control. There are currently no commercial GM crops, although all imported food and ingredients derived from GMO must be approved by the food safety authorities and clearly labeled on the packaging before sales. Criminal and civil penalties may be applied in relation to breach of these legislations, and offenders may be ordered to mitigate or remedy any adverse effects on humans and the environment. Thank you.